I now have the pleasure of introducing Dr. Laura Miller, who's the keynote speaker for the conference. Dr. Miller came to K-State in 2023. She holds the rank of Associate Professor in Veterinary Virology. She had her training started with a Bachelor of Science with honors in medical microbiology from Edinburgh in Scotland, and a PhD in molecular virology from the Institute for Animal Health at Pirbright, which is conferred by the University of Reading in England. She served as leader of serological testing at Pirbright during the UK foot and mouth disease epidemic of 2001. And then after that, Dr. Miller must have had enough of Pirbright and moved to the US to complete a postdoc as a research associate, and then was employed for 3.5 years as a tenured research microbiologist at the US Meat Animal Research Center in Clay Center, Nebraska. She then moved with USDA Ag Research as a microbiologist to the National Animal Disease Center in Ames, Iowa. She currently serves as past president of the American Associations of Veterinary Immunologists, member at large of NC 229, Multi-State Committee for the Detection and Control of Porcine Reproductive and Respiratory Syndrome Virus and Emerging Viral Diseases of Swine, collaborating principal investigator in the Center on Biologics Development and Evaluation, and a project director, NSRP 8 National Animal Genome Research Program. No wonder you don't have time to do anything. Dr. Miller's research focus has been on virus-host cell interactions and application of this knowledge towards a better understanding of the pathogenesis and immunology of viral pathogens. She was the first to report on this essential role of the cytoplasmic domain of the alpha V beta 6 integrin in endocytosis-mediated infection by foot and mouth disease virus. She has used and adapted evolving technologies, including microarrays, serial analysis of gene expression, digital gene expression tag profiling expression, RNA-seq, SNC RNA, STOMICs, prediction matrices, cell lines, and organoids to conduct basic and applied research that has characterized the cellular response to viral pathogens. Recently, Dr. Miller's host response studies have progressed to transcriptomic analysis in air-liquid interfaced respiratory epithelial cells, resulting in the creation of the most extensive immunoinformatics dataset to date on host gene expression in response to porcine respiratory viral infection. Dr. Miller continues to identify the mechanisms of virus-host interactions and viral pathogenesis, improve diagnostic methods and vaccine strategies that will aid in the control of emerging and re-emerging infectious diseases. Dr. Miller, please awe us.
Thank you, and I'd like to thank the organizers for inviting me to give a keynote presentation on some of my research and stepping back into biomarkers and explaining the role of transcriptomics. And I'll try and reflect that on some of the biosafety and biosecurity and biodefense that's needed. So what is a biomarker? It is really, could be anything from your blood pressure to a molecule that we can measure as a phenotype for a disease. It's been classified also in chemistry and toxicology markers for biological processes, pathogen processes, or pharmacological responses. And it can be any substance, structure, or process that can be measured. And it's mostly a measurement, rather than an endpoint looking at morbidity or mortality, it's a surrogate endpoint during a biological system. So with advances in transcriptomics or sequencing, where we have large DNA sequencers and actually decreasing in size, as we can see here, with potentials for sequencing, we are getting more sequences. It's getting cheaper to process these sequences, and we get larger databases. And with that, we need faster technology for tools for analyzing this. And so we can see the decrease in cost for the human genome that's since 2000 has accelerated and decreased in cost, and acceleration in sequencing and decrease for raw megabase DNA sequence. So with these advances, we can look at a variety of things in pathogen characterization surveillance, in the host and vector pathogen interactions, in population genomics, so susceptibility, but also population genomics when we're talking about feeding the world, and the best improvements we can do in agriculture. And then in microbial genomics, so in resistance and AMR or antimicrobial resistance, the metabolism and immune responses, as well as nutrition. So there's a number of transcriptomics that we can look at, mostly using next-gen sequencing. But not only when you're looking at transcriptomics do you just think about the transcriptome and transcription and RNA, you're actually got to think about the whole picture. So genome, proteomics, interactome, metabolism, and into the phenotype that you see with disease. So when you're thinking about the interactome, you're thinking about that systems biology or network of networks, including down from the genomic or single cell level, or even small non-coding RNAs, all the way up to the animal and the clinical phenotypes and all the other influences that can be on there, the proteomic network, the metabolomic network, the social interactions or environment, and then environmental exposures and microbe interactions. And then thinking about all of these will improve our understanding of pathobiology, classify the disease, and then we can do network targeted therapies. So looking at the interaction in this interactome throughout. Also using new technologies in artificial intelligence, we can look and take this vast amount of data and towards precision medicine, and we can then look for gaps and where we can target more clearly. I'm putting in a little bit about pathogen genomics, although I'll probably be focusing more on the host genomics in response. But in pathogen genomics, there's a number of artificial intelligence and machine learning technologies required in this. When you're doing next generation sequencing, looking at that genome, doing bioinformatics analysis and phenotypic tests are the classical and previously used. And now we can use machine learning with unsupervised models and supervised models to inform us on the genotype and the pathogenicity outside of normal techniques that were done in the lab. And therefore we can predict virulence, we can predict host susceptibility, we can predict the genotype, strain, or subtype that would be, or variants that are coming through that would be, we could be susceptible to in a potential pandemic. And we can also look at pathogenicity and our potential therapeutics or vaccines or antivirals. So key challenges in this is that transcriptomics data can be very complex and it's difficult to analyze. There's advanced computational methods that are required to get some meaningful patterns in gene expression. That's kind of how we look at this data analysis. We're profiling what's being expressed to identify targets and candidate genes to target with antivirals or therapeutics or vaccines. Important is also sample collection. I'm not going to go into depth today on the importance of how we collect our RNA or materials, but particularly in the context of biosecurity, when you're taking some virus out, an RNA, what are the best methods to be able to collect those samples and get a good idea of not only spatially, but temporally and kinetically with those samples as well, and the best collection for that. And then we need to standardize that sample collection as well. And so this is important for processing and analyzing to ensure that that data can be compared across different studies. And these are important for screening and genetic containment. So for an outline of what I'm going to talk about today, I'm going to give you an overview of some basic transcriptomics that we have had done previously, and then I'm going to talk about some advances in transcriptomics, and I'm going to give you an example of a study where we were using transcriptomics for a biomarker discovery with SARS-CoV-2 in COVID disease. So I'm going to step back in case we're not all biologists in the room or online. So why do we measure RNA? Why do we choose RNA? In the body, there's more water. So why not water and lipids? Well, water is everywhere. There's not much differences there. Lipids are difficult to work with. Why not the DNA? Well, the DNA is constitutive throughout cells. You're always going to have DNA. RNA is more measurable, and you'll see differences in expression. Why not proteins? Well, there is proteomics, but they're not giving us as much information as RNA would. And they're later when we want to look at an early response. So why are we looking in cells and tissues versus in tubes? Well, we want it in the spatial or in the organ or in the target region that we know what's going on exactly where we're looking at these genes. And why high throughput? Well, the more information you get, the more we can define and find out more. And so with genes, how many genes are in the genome and how many cells are in the organ? So with that central dogma, we're looking at RNA rather than DNA. And then we can also look at protein. When I said we can look at that overall look at transcription, translation to the phenotype and their interactome beyond that. And so at the cellular level, we can look at gene expression and use sequencing technologies such as bulk RNA-seq. But for the spatial level, previously we used FISH, or fluorescent in situ hybridization. Now there's new technologies where we could use single-cell RNA-seq and spatial transcriptomics. So bulk RNA-seq, or using microarrays as we did about 20 years ago, we still do for high throughput if you know what genes or exact RNAs you want to look at, so you can use a micro array. From that, we progressed forward to SAGE, or serial analysis of gene expression. This is very useful because it allowed you to look at unknown transcripts and collect a lot of data at the time when genomes were not annotated very well. And then later we moved on to digital gene expression tag profiling, which allowed you to do transcript abundance in a better measure. And this was all prior to now RNA-seq is the standard for this kind of transcription analysis. So when you're looking at bulk RNA-seq, you're looking at the whole organ tissue or cells, if you've collected cells of whatever you've got. And it's kind of like you've got all this fruit in a smoothie because you've got a bulk amount of information, or a whole lot of paint and a painting, but not really any space or telling us anything about the tissue sample it came from. It's just telling you the genes expressed in that tissue sample. The technology and methods we developed for doing bulk RNA-seq, we've developed for pig blood, whole blood, but I've also done it with tissues, lymph nodes, cells, a lot of different ways. This one was quite nice we did because we were able to look at mRNA and small non-coding RNA in the same sample. So this was quite a new technique. And we reduced the globin, which would interfere with how much information we could get. And then we developed the transcriptomic analysis from free open source software. And that's constantly building on that, but it was kind of a nice thing that we put together, a nice pipeline where we can look at this differentially expressed gene data. And from that, we'd established a method and a training guide for examining networks. So from gene expression data, you start to look at networks, how they all interact, those genes. And then from that, you look at pathways and gene ontology terms so that you can classify all that data that you're getting. And that will give you a little bit more information, how these genes interact, which one would be your targets. So my example of this work we did right at the beginning of SARS, and before we were even talking about host susceptibility of other animals, and we just thought it was humans. I did some prediction work with the colleagues at Tennessee State University, and we predicted that the white-tailed deer would be susceptible to SARS-CoV-2 infection. And not long after, surveillance was done, and it was found that there was SARS-CoV-2 in white-tailed deer. And that was detected in 12.2% of the deer when they did a survey of 11,000 on the eastern of the United States. And then when they did more surveillance, we actually found that there was 31.6% had antibodies. And interestingly, also the exposure of these animals and the antibodies and the variants that we saw followed that human exposure and then what was going in the human population circulating. And that we saw that SARS-CoV-2 was transmitted, at least with these variants, that there was 106 times from humans to white-tailed deer. But while this was happening, we saw that there was no studies being done on the innate immune factors and why with deer we see no disease. And with human, we see this cytokine response. We see this long COVID, we see illness to COVID, but no, there's high transmission of SARS-CoV-2, but no disease in the deer. So we decided to look at primary respiratory epithelial cells and compare them between human and deer infected with the Washington strain of SARS-CoV-2 at an early stage of infection to see if potential biomarkers, because this could give us clues why, when we see no disease in deer and we have asymptomatic humans, maybe that would give us some idea of why some people, groups and subgroups of population were asymptomatic. So the methods for doing this was we collected the respiratory tract from some white-tailed deer. We digest those epithelial cells and seeded to make a primary respiratory epithelial cell culture. These grew well in comparison, and similarly to the human, they were possible to infect with SARS-CoV-2. We saw CPE within 48 hours. At an MOI of 1, so, multiplicity of infection of 1. And we saw that there was CT values. We could see that there was replication of the virus in human and deer. We could compare treatments over 48 hours. So both deer and human respiratory epithelial cells were susceptible to SARS-CoV-2 infection. And from that, we collected RNA-seq to measure that differential gene expression. And that's with RNA from the samples, Delegate, do next-gen sequencing and Illumina sequencers. And then we mapped the reads, did a huge amount of data analysis, and then looking at statistical analysis, and then biomarker identification, pathway analysis, and the interpretation of this data. So what we did see when we looked at the overall digital gene expression data from human versus deer infected, the humans decreased in the number of genes that were being differentially expressed over that 48 hours, while the deer increased in the number of genes differentially expressed over the 48 hours. And while we didn't know many of the genes, because the deer genome has not been well annotated, I don't think it's even public yet, we had to go back and re-annotate the unknown genes that were differentially expressed and of interest. And so we were able to look in comparison, looking at upregulated differentially expressed genes. And one of the ways to get that overwhelming amount of data on genes is to compare what's shared and what's unique between those time points and between our human and deer, so we could identify the genes over time, and then what was downregulated as well. So then we looked at what pathways were enriched between the two and how many of them and how many were shared pathways over that time. And we found that one of the pathways early at 6 hours was the IL-17 signaling pathway. At 24 hours, shared pathways IL-17 signaling for differentially expression, and the pathogen-induced cytokine storm, which is kind of what we know with humans and COVID, that there's a cytokine storm signaling. And again, we saw that at 48 hours. So when we went into depth and looked at these pathways at 6 hours, you can see just by color, which indicates where we see in orange activation predicted, inhibition is in blue, and those important genes are magenta of importance. And we're looking at downregulated genes are green, upregulated genes are pink. We can see the relationships between these genes and these pathways, but we see a stark difference between human and deer at 6 hours in the IL-17 signaling pathway. And when we have a closer look, we actually saw that this was particularly in the HSP90 and the TRAF genes, where there wasn't a difference in this in the deer. And when we look further back and we look at cytokine signaling, we see downregulation of the CXCL genes, whereas there was upregulation in the human towards the June response. When we looked at 24 hours, and this is addressing the cytokine signaling pathway, we saw differences with the deer at 24 hours in down modulation again, whereas we see some upregulation in the human at 24 hours, but mostly going less downregulated. And when we look at 48 hours, that switched to upregulation. So that's where you're seeing that differential expression in the human, and that large number of genes switch is from inhibition to activation at 48 hours, but continues to be downregulated in the deer at 48 hours. So we can see at 48 hours the difference between those two. So this, to summarize, the white-tailed deer were susceptible to SARS-CoV-2, the cells, but they didn't cause a hypercytokine gene expression, as we see with the human. And then a downregulation of that IL-17 and NF-κB pathways, which are very important in innate immune responses, leading to any other adaptive responses. They could be the key to regulated cytokine response in the deer cells. So maybe the clue of why we're not seeing that disease in deer versus human. And that means that they could play a critical role in an early antiviral and tissue repair response, and almost giving us a model there of deer for SARS-CoV-2 infection that helps us identify things for the human responses and targets. So with advances in single cell or advances in transcriptomics, we now have single cell RNA-seq, where you're looking at, rather than that big tissue sample or group or that smoothie, we can look at the individual fruit and isolated cells. So this is more of a cell typing technology, but telling us the gene expression within those cell types. So you're being able to break it up a little bit more information. So that when you're doing single cell, you can do tissue dissection. You basically are single cell sorting and sequencing, and that gives you clustering and cell type identification. So the problem with single cell, and what a challenge is you're really cell typing and not really giving a spatial effect of where it is in the tissue or the organ. And so you're kind of getting a little bit of a fuzzy picture, and there's insufficient spatial resolution. So how do you clear that picture? Well, we use certain kinds of deconvolution computer tools to identify those cell types and give us a little bit more information in single cell RNA-seq. So this is a collaboration I have with a colleague at Brown University. She's developed the spatially informed cell type deconvolution for spatial transcriptomics, bridging from single cell RNA-seq towards spatial transcriptomics. So you can look at that expression data and get a little bit more information. Still not as clear as we quite like. You're targeting your cells, you're telling your organs. So now we have newer technologies where we're actually looking at the fruit in a spatial 3D, like as in a fruit pie. So where you've taken off rather than groups, you're actually getting those individual cells and where they are in the organ. or where they are expressing. So there's a lot of, as I said, deconvolution type technologies. We now have a lot of platforms for doing that kind of analysis. What we have now in currently in our labs, we have this new geomics for looking at spatial transcriptomics and at the region of interest level. And then we can do all these computational, there's CARD, which my collaborator has. And then we also have the ability to now look at this at a single cell or subcellular level with the new CosMx. So we advanced from that single cell to spatially resolved single cell using the CosMx from NanoString. So these two technologies, the geomics and CosMx, which we now have in the College of Veterinary Medicine with CZID, USDA sponsored that geomics, and then CosMx was awarded to me through the NSF Engines Biomanufacturing Training and Education. So part of the core, we are able to look at using the geomics, which is a digital space profiler. We can look at tissue structures and whole transcriptome. And then with Imager, which is the CosMx, which is a spatial molecular imager, we can look at the single cell and subcellular level. So these are both machines funded through CZID, USDA, and NSF Engines, but they require quite a workflow to them, which I'll go into a little bit. One of the things that's nice about these is they complement each other. As I said, you can look at RNA plus protein on the same slide, and that gives you very accurate data. As well as cell type, you now can do cell segmentation. So rather than looking at those clump or that circle or that grid pattern and that fuzzy picture, you can now look at the individual cells because you can segment them. And then you can look at the RNA expression within those cells and protein expression. So it's quite a lengthy workflow for these. One of the advantages talking about biosecurity-wise is we use formalin-fixed paraffin-embedded tissues for these. So that means we can bring out BSL-3, BSL-4, potential tissue samples and use these and look at these at a spatial transcriptomics level. So for these, we have to then make special slides from those tissues, incubate. There's quite a few steps, but now we have it almost semi-automated with a Leica semi-automated stainer for the protein level. So we stain and wash those slides and run them. And then we have intensive data analysis on the geomics. We're looking at whole transcriptome. And then on the CosMx, we're looking at probes or reporter genes at, we can go up to a 6,000 plex level. So very high reactome plex. And this, why would plex matter? Why do we want to go that high to 6,000? Which quite, so normally on a normal panel that's offered about 247. This is about the amount of information you're getting. When I was showing you those pathways and really narrowing down those pathways, this is kind of where you're looking gene ontology. When you go up to 1,000 plex, now in green, these are the things that you can discover at 1,000 plex. When you go to 6,000 plex, you've got very few gaps in that information. So you're getting a lot of information. And so you can do this all in a single slide. You don't need to do the FISH. You don't need to do the single cell RNA-seq. So this is very nice with the NanoString CosMx. So you can use the formalin-fixed tissues or fresh tissue sections. It's not destructive to the RNA. So you get RNA and protein profiling. And then you're doing that single cell. It quantitates up to 6,000 genes. And then you're getting cell-to-cell. You can look at cell-to-cell interaction, ligand receptor interaction, and nearest neighbor analysis. So what is cell-to-cell interaction, ligand receptor? That is where I'm hoping to use for targeting my antivirals or my vaccines so that I know exactly which cells I'm targeting to. Another way we're using this is for cancer or oncology. We can now look at what's healthy tissue, what's diseased tissue, or healthy cells versus diseased cells. We can look at infected cells for virus, uninfected. We can use it for chemical, toxicology. There's a lot of opportunities there for looking at the cells in their own in situ, so where they are and comparing them to the cells around them. and what genes are expressed. So again, a lengthy workflow, but a lot of data that you get out. And currently we are creating pipelines for doing analysis for different pathways. So we get a lot of information when we create these pipelines where we can look at our cell typing and our marker genes at that level. Other uses for me, of course, I'm doing a lot of animal genome research. So I'm working with FANG, which is the Animal Annotation Genome Consortium, where we're getting a little more genome information. So again, I'm looking on the host side. We can look on the pathogen side and also make probes towards the pathogens. So towards that, this would steer us towards potentially gene editing of animals, because we'll find which genes we really want to target. We can look at which tissues. And so there's a lot of requirements coming from the field, from the agriculture, for this kind of information at this level. And so I will take questions for time.
Questions for Dr. Miller?
Yes.
Is the Pathobiology up and running now?
It is. We are doing our first analysis of data. We've done some PERS and in lung, and we've done some ASF in tonsil. We have other plans. I have a long waiting list of people wanting to run things. We mainly have the panels built for human, universal panels, and we have mouse. There's a canine consortium, but that's more geomics. So I'm developing panels for different species and adapting what we have for human. We have a lot of cross-hybridization as well. So that's the good thing, is what we've run already was with the human universal panels, and we're getting very good results and good numbers of transcripts. I didn't put that data up, but we did get a good number. Yes.
Applications for the ranching, farming, ag world that we're living in right now in Kansas?
All sorts. So I have ones from like the Water Institute. So when we're looking at environmental sampling, we can even look at cells for that. So at all different levels, we can, agriculture-wise also, well, I mainly work on diseases for agriculture. So like PERS, where we're looking, they've already developing gene-edited pigs. So we can find a little bit more, and they're also working on genome for resilient pigs to multiple diseases. So for agriculture, that's kind of a very important line of where are we going with breeds and where are we going with gene editing, but also potentially therapeutics, diagnostics, vaccines for that. So getting more targeted vaccines. So that's one of my lines is a prototype vaccine that I'm working on with an antiviral involved. Yes.
I know that there was a talk of possibly COVID spreading the deer from farm deer, so humans to farm deer, then farm deer going to wildlife deer. You guys testing farm deer, or
these were mostly wild deer, because this is a surveillance like they do for CWD, a chronic wasting disease. So this was a general surveillance, but also they did do a specific surveillance as well. Mostly now, and I don't think it's been said very definitively, but it looks like it came from the water plants and wastewater. That's the human transmission to the deer. There have been cases of deer to human now as well. So that was one of the important things, is wildlife being a reservoir for diseases and spillover. And that's one of my big interests in prediction of susceptibility, is those spillovers events happening, and that prediction that we can do.
Is the deer to human transmission mostly during butchering?
No, it's, I think, a handling, because it's aerosolized. But that's because of that. Yeah, but it's only been like 1 or 2, and it was a variant, and it's still a little bit questionable. But I mean, we're seeing it now with H5N1, so that's part of the things to look at as well.
How are the deer actually getting infected?
I think it was from the wastewater. So I mean, that's weird, because I just said it's aerosolized normally. But I think just a coronavirus epithelial, that high virus load that was there.
That's RNA in wastewater. I mean,
Yeah,
How much infectious viruses do you have access to?
and just being that close, it would be enough that, I mean, that's not my, that,
It's not like deer are in the sewers.
no, but they were, I mean, where the location, so most of that data comes from University of Penn State, actually. I think they did that in Pittsburgh. So they sampled round and it looked like they were localized in urban areas more.
Yeah, that's interesting. So there's still the older storm sewer systems that when there's overflow, you have storm water and there's preponderance of before it gets to the wastewater treatment plant. I could see where that would be a higher density in the urban setting because of the older systems.
Yeah.